Hello, everyone. Welcome to the session on connectivity in health and disease. I'm chairing the session. I'm Stephanie Volker with Ruby Kong next to me here. And we are very delighted to have 5 of our speakers here with us today, and one presentation will be a video. Now, the first speaker of the session is Stuart Oldham, who's going to talk about the conserved developmental access of thalamocortical connectivity in humans. Stuart.
So I just want to say firstly, thanks to the organisers for letting me come along to present some of our work in characterising thalamocortical connectivity. So without further ado, the thalamus is a subcortical structure which has widespread connectivity to the cortex. This widespread connectivity facilitates communication from the peripheral nervous system to cortical regions, as well as communication between cortical regions themselves. And this gives the thalamus a key role in shaping whole brain dynamics. Therefore, if we want to understand whole brain dynamics, we need to understand the organisation of thalamocortical connectivity. Many studies looking at the organisation of the thalamus focus on its nuclei, which are approximately 30 regions which have distinct psychoarchitectural properties. However, the principles that underlie the patterning of cortical projections from these nuclei remains unclear. This can be seen if we cluster areas of the thalamus by their structural connectivity. So we can see that the boundaries of these clusters do not neatly align with the boundaries of our nuclei. And we can also see the same occurs with functional connectivity. And so what we need is some principle which explains how these patterns of connectivity vary both across and within nuclei. A paper a few years ago conducted in the mouse attempted to do this. So this paper by Phillips et al. Found that thalamic nuclei could be ordered along a medial lateral axis of gene expression. And furthermore, they found that position along this axis was associated with differences in the properties of cortical projections, suggesting that this axis represents a fundamental organizational principle of thalamocortical connectivity. So we wanted to see whether the results obtained in this mouse model would extend to the human model as well, as doing so would provide further evidence of this principle. To do so, we first, throughout the thalamic volume, defined a set of thalamic seeds. And then using data from the Allen Human Brain Atlas for 2,000 genes which have been shown previously to have above average expression in the human brain, we mapped each of these to each seed, constructing a seed by gene expression matrix. Then, because we also wanted to look at connectivity patterns from the thalamus, we also conducted probabilistic tractography from each region to 250 cortical regions in the left hemisphere. And by repeating this across all seeds, we can construct a seed by cortical region connectivity matrix. And we repeated this across 76 participants of the Human Connectome Project to get the average matrix you see here. And because this is normalized in the same way as our gene expression matrix, we can combine it with this. And so we get both the seeds' connectivity patterns across cortical regions and gene expression. And we want to extract gradients from these. And so to extract a gradient from this, we can use everybody's favorite gradient extraction technique, which is, of course, principal component analysis. So principal component analysis returns us two matrices, a matrix of principal component scores, which tell us how strongly each seed relates to the underlying component, and principal component loadings, which tell us how strongly each gene and cortical region defined each component. And we can use these to answer the following questions. Does a dominant medial lateral gradient occur in the human thalamus? And if it does occur, what kind of projections emerge from it? So returning to our principal component scores for a moment, we only want to extract those that are related to the first principal component, namely the one that explains the greatest degree of variance, in our case 30%. If we take these principal component scores and we can project them onto their respective seeds and surrounding voxels, we see a medial lateral gradient emerge. And so for those of you not living in gradient world or have forgotten how PCA works, you might be wondering, this is very pretty, Stuart, but what does it mean? So basically, a region that has a high principal component score on variables, so namely cortical regions or genes, which have a high loading on this particular component, they have a high original value. And when it's a negative loading, they have a low value. And for regions which have a negative score, this is reversed, so now there's a negative association. So beyond this visual inspection, we can also directly correlate each seed's medial lateral position against its principal component score, and as you can see, this further confirms that what we're seeing is indeed a medial lateral axis. So we have this medial lateral axis, but we want to know if it corresponds to what was observed in the mouse. And what we can do is examine if a similar set of genes are associated with each of these axes. And to do so, we can use our principal component loadings for the genes that we defined earlier. And what we can do is we can run an enrichment analysis. So in this case, we are asking, are the genes which are most strongly associated or positively associated with our medial lateral axis enriched for genes in the mouse? And indeed, what we find here is that the genes which have the largest positive association with the medial lateral axis in the humans are also enriched for genes which have the strongest loading in the mouse. If we look at those with the strongest negative association, we see that they are indeed enriched, although not to a significant level. However, if we take the principal component loadings for the mouse genes and plot them against the principal component loadings for the human homologs, we see that this is indeed a positive relationship, which overall this suggests that a similar set of genes are defining each of these axes in the mouse and human, suggesting a conservation across species. Finally, we wanted to look at cortical projections from this thalamocortical gradient. The first way we can do this is if we plot the connectivity pattern from the seed with the highest score and we plot through to the lowest. So when we do this, we see that the pattern of cortical connectivity moves from posterior sensory regions towards more frontal anterior ones. We can also look at a gradient, this cortical projections, by taking the principal component loadings for the cortical regions and projecting them onto the brain. And when we do this, we also see this posterior-anterior gradient emerge. And so what this is telling us is that medial regions in the thalamus tend to preferentially connect to anterior regions in the cortex, and lateral areas in the thalamus tend to preferentially connect to posterior regions in the cortex. Also from the results in the mouse paper, it suggested that thalamocortical connectivity is associated with distinct electrophysiological profiles. And previous work looking at functional thalamocortical connectivity has found that this is associated with cortical intrinsic timescales. So an intrinsic timescale indicates whether a particular region's intrinsic neural fluctuations tend to be fast, indicated in red, or slow, shown in blue. And as you can see visually, this looks like quite a close match. And indeed, when we correlate these two gradients, we indeed see that they are very strongly related. So our cortical projections are very closely reflect intrinsic timescales. So to summarize what we've just seen, we find that there is a medial lateral gradient in the human, and this seemingly has reflected and seemingly is capturing what has been observed in the mouse. And also we find that this gradient projects to distinct cortical patterns and is associated furthermore with key functional cortical patterns. It's also interesting to note that these two thalamic and cortical gradients correspond to fundamental axes of development. So in the cortex, neurogenesis begins in anterior regions and progresses to posterior regions, and in the thalamus, it begins in lateral regions and progresses to medial regions. So this suggests the properties of these gradients potentially emerge due to this early developmental event. It's also interesting to note that regions in the thalamus, which begin neurogenesis first, connect to regions in the cortex, which begin neurogenesis last. So this suggests there might be some interplay between the timing of development, which causes these thalamocortical circuits to emerge. And so to summarize what we have all learnt today is that we find that there is a dominant medial lateral thalamic gradient informed by gene expression and cortical connectivity. This gradient is seemingly conserved across species. We also find that this gradient maps to a distinct cortical patterning and is associated with distinct patterns of cortical function, and this gradient may also reflect sequential development. And so with that, I'd like to end it there. If you would have any further questions beyond in this session, you're welcome to come and see me at my poster today and tomorrow. I'd also like to give a quick word of thanks to Dr. Gareth Ball, who really helped to conceptualize this project and indoctrinate me into the world of the gradient, to Dr. Irina Arnaikevichude for helping me move through the world of imaging transcriptomics, to the Human Connectome Project, Allen Institute for Brain Science for providing all the open access data that we use in this study, to the funders of all this research, and finally to all of you for coming along today and listening. Thank you.
So apparently we got a lot of questions based on how do we make these animations like that, or which software you use to make this fantastic presentation, and how much time have you spent? So, right, and yeah?
So I'll answer them. So in regards to the amount of time I spent, so either it took a really long time for me, in which case I look bad, or it really took a really small amount of time, in which case you will look bad. So I'm going to refrain from answering that question too closely. But in regards to what I used to actually make it, believe it or not, about half of it is actually done in PowerPoint itself. There's a surprisingly deep layer of complexity to PowerPoint, which I strongly encourage you to all explore. And otherwise, all the animations are made with the famous filmmaking software MATLAB. So, and then it's all just combined with a lot of editing trickery to make it all look seamless.
Okay, so for the real science question, the first one will be any speculation on if this reflects metabolic processes, cell types, or other features that underlie brain activity.
So repeat that again, sorry?
So any speculation on if this reflects metabolic processes, cell types, or other features that underlie brain activity.
So we haven't explored it too extensively, but looking at the genes which are most strongly associated with this particular axis, they seem to be related primarily to neurotransmission, but we haven't really extensively explored what kind of underlying mechanisms may be driving this gradient. I mean, I presented some initial speculation based on what we see with this corresponding to axes of neurogenesis, but that is very much the next step that we're going to be looking into.
Okay, thank you. Okay, one more question. Can you comment a bit on the specific processes implicated on your observed gene sets?
The specific process, so as I just said before, that we haven't looked at it extensively, but it tends to be genes related to neurotransmission.
Okay. The last question will come from me, actually.
Okay.
So when you Yeah. So when you apply the PCA on your like a concatenated gene expressions and connectivity, did you observe any different like a PCA1 score if you only apply the PCA on the gene expressions without anything from the other information?
Yeah, so it's a really great question. So we spent a lot of time on this. So trying to figure out, like thinking of, because obviously there's a lot more genes than there are cortical regions, which could sort of bias the PCA. And so we've tried restricting the amount of genes to only those with the most differential expression, only those which are mouse homologs. We've tried taking PCAs of the cortical connectivity matrix and the gene expression matrix and combining that and then doing a PCA on that. And every single time we see pretty much the exact same gradient emerge. And if we do the PCA on just the gradient, the gene expression, we also see the same gradient. If you do it on just the cortical connectivity data, you don't quite get the nice pretty gradient we saw before. There is quite a strong anterior-posterior gradient in the connectivity data as well, and so the first and second PCs for just the track data tend to be sort of a hybrid of a medial-lateral anterior-posterior.
Thank you.
For those of you who want to see those beautiful animations again, he actually provided a lovely video online that you can just find on the OHBM channel. Right, we're moving on to our next speaker, who unfortunately can't be with us today. That's Pin-Nong Chen, but we're going to have a video with the title Altered Global Signal Topography in Alzheimer's Disease Correlates with Neurodegeneration Pathways.
Hello everyone, my name is Pin-Dong Chen. I'm a PhD student in the Institute of Automation Chinese Academy of Sciences. It's my honor to have a chance to share my study, Altered Global Signal Topography in Alzheimer's Disease Correlates with Neurodegeneration Pathway. Alzheimer's disease is a typical neurodegeneration disease associated with widespread disruption in different brain areas through disconnection mechanisms. Those widespread disruption breaks the balance between local specialization and global integration in the brain, and consequently impair the daily cognitive ability. The global signal GS refers to the average signal of the gray matter or the whole brain. The GS is suggested to link with the artifacts of head motion, hardware, respiratory, and other unknown effects. Therefore, it was regressed out as a non-neural signal in pre-processing of fMRI. However, there are still some controversies on whether to regress the GS in fMRI. Recently, some studies suggested that the GS is not merely contain noise. GS could represent an overall fluctuation of the global activity, and the correlation between GS and the local time series could represent a local contribution for GS. The GS were associated with task modulation, weakness, and disorders such as schizophrenia and autism spectral disorder. But whether the disrupted local global integration in AD impaired the GS and the GSLR is not clear now. Moreover, we should note that the GS is associated with artifacts. How could determine the changes of GS truly reflect the changes of AD is a problem. Fortunately, we have the imaging transcriptomics. Imaging transcriptomics provides a novel perspective to explain the neuroimage. It could recover the underlying mechanism of the alteration in neuroimage based on the spatial association between transcriptomics data and the neuroimage. So we introduced the imaging transcriptomics to investigate the association between the GIS underlying mechanism and the AD. There is a dataset used in the study. We utilized a large sample of an fMRI dataset from the multicenter Alzheimer's disease initiative, including 257 normal controls, 257 mild cognitive impairments, and 295 AD from Stephen datasets. This is the pipeline of this study. First, the mean time series of gray matter was used as the J's time series, and we calculate the Pearson correlation between J's time series and each voxel time series in the brain. The Pearson correlation map is the global signal correlation map, the GSLR map. We obtain the GSLR map in each group and get the alteration map by group comparison using a general linear model with the gender, age, and dataset as covariables. Then we used the Brainotome atlas to obtain the regional GSLR alteration, and the partial least square PLS regression was applied to investigate the association between GSLR square, GSLR alteration, and the transcriptional data from any human brain atlas. Then we selected the significant genes by permutation, and genes were ordered by the PLS weights. Finally, we applied the enrichment analysis on the significant genes list by the Kyoto Encyclopedia of Genes and Genomes, the KGG. Next is the alteration of GSLR. There is a bidirectional change pattern in AD. The increased area were mainly located in the frontal lobe and the inferior parietal lobule. The decreased area were mainly involved in the temporal lobe and the cingulate cortex. After the FDR correct, the increased areas were mainly involved in the superior frontal gyrus and the middle frontal gyrus. The decreased area were mainly involved in the superior temporal gyrus, lateral occipital cortex, and the hippocampus. Here is the results of the cognitive relevance of GSLR. We extracted the mean GSLR in the decreased area and the increased area. GSLR changes in those regions correlates with the cognitive ability and demonstrates a progressive trend from the normal controls to Alzheimer's disease. The decreased GSLR showed difference between groups and positively correlates with the MAC score, and the increased GSLR showed differences between groups and negatively correlation with the MAC score. Next is the results of the imaging transcriptomics. This is the PLS component 1 score spatial distribution, which is correlated with the GSLR alteration. And we selected the genes had a weight score bigger than 3 and then performed the KGG enrichment. The results of enrichment analysis include several pathways that associate with neurodegeneration disease such as Alzheimer's disease, Huntington's disease, and Parkinson's disease. So in conclusion, our study begins with the AD phenotype. We finding a bidirectional change pattern in AD, and the change pattern showed correlations with the cognitive ability. Next, we found the genes which is related to the GS spatial alteration patterns. Finally, we found the background molecular mechanism of those genes are associated with Alzheimer's disease. Our study highlights the relationships between local activity and the global activity that is changed in ID, and we proved that the informative role of GS in AD. Finally, we provide a new perspective for understanding the functional system changes in AD. In the end, I want to thank my collaborator from the Multicenter Alzheimer's Disease Initiative and my faculty in the Institute of Automation Chinese Academy of Sciences in Beijing University of Posts and Telecommunication and Beihang University. And I also thanks the grants that supports our studies. Thank you.
So obviously we can't answer your questions on this one, but feel free to contact him via email if you have questions on this talk. We're moving on to the next one. Our speaker is Ahmed Samara, and he's going to present his presentation on gradients go to the movies, macro-scale cortical organization during naturalistic viewing.
Hi, I'd like to start by thanking the organizers for giving me the opportunity to talk about our work here. And I'm going to be talking about the macro-scale cortical organization using functional connectivity gradients during naturalistic viewing. I'm going to start by providing a brief background on how this project came to be, and then describe the data we used and the analysis we performed, and present the results of the gradients during movie watching as a naturalistic state, and how we try to relate that to behavior in a meaningful way, and then provide some conclusions and future directions. So this project actually combines two emerging fields in neuroimaging research. The first is naturalistic imaging, or naturalistic paradigms, which are studies that use complex stimuli, such as movies, to approximate real-life scenarios and improve the ecological validity of these studies. And the second field is gradients in brain organization, which is a method of dimensionality reduction that seeks to describe how certain features in the brain, such as functional connectivity, changes across the cortex in a gradual manner, instead of within regions with hard boundaries between them. And you might be familiar with Daniel's gradient, which describes an axis from unimodal to heteromodal brain regions that was based on resting state fMRI data. So we became interested in the following question. Did these functional connectivity gradients, or this way of describing the organization of the brain, evolve to support the kind of cognitive processes we see during resting state, or during the more complex processes that we observe during naturalistic paradigms? To answer this question, we use data from the Human Connectome Project 7 Tesla data release, which includes data from healthy young adults who are scanned in 4 resting state and 4 movie watching data runs. And during the movie runs, subjects watched 4 to 5 movie clips, and there was 20 seconds of rest between or before each clip. So one thing we know about movie watching as a brain state is that we get, as an acquisition state, is that we get significant differences in head motion compared to rest. So we wanted to make sure that we account for this effect and mitigate the effects of head motion on our analysis, since this is the first time we're describing these gradients during movie watching. And we started by excluding any subjects with a head motion, with a mean frame was displacement above a certain threshold. And then we implemented a motion censoring process where we applied even a higher threshold for head motion. And we tried to optimize comparisons across conditions by removing the rest intervals within the movie runs and making sure that we're left with a comparable amount of data per run for all subjects. So for the analysis itself, we started by concatenating all 4 runs within each condition and averaging the time series of vertices based on their assignment to 1,000 regions from the Schaeffer functional parcellation. And then we computed a single functional connectivity matrix per subject per condition. And we also computed a group average functional connectivity matrix per condition. So to characterize the functional connectivity gradients during movie watching and compare that to rest, we used the BrainSpace toolbox and to compute separate sets of functional connectivity gradient for rest and movie. And this is just a conceptual representation of the analysis. We started by our group average connectivity matrix in rest and movie, and then we applied a cosine similarity function to compute a new affinity or similarity matrix that was then decomposed using the dimensionality reduction method diffusion embedding to get gradients that are ordered based on the variance they explain. And I don't have the cool animation that Stuart have to convince you that this is the best, or this is everyone's favorite dimensionality reduction method. Okay, so if we just look at the variance explained by the top gradients in movie and rest, we see that it's very similar across conditions. But when we correlated the top gradients in rest and movie, we found that gradient 3 in movie was not correlated with anything in rest, which might indicate that it's capturing an axis that is inaccessible during rest. So when we look at the topography of this gradient by projecting the gradient scores on the brain surface or the cortical surface, our rest gradients nicely replicated previously described gradients. And when we look at the movie gradients, starting by gradient 1 in movie, we see that there's similarities in the overall organization of this gradient with some important differences. For example, we see that visual cortex is shifted along this gradient into intermediary way between unimodal and transmodal and heteromodal regions. And then we see that auditory regions are no longer grouped with the somatomotor network at the unimodal end of this gradient. If we look at gradient 2 in movie, we see that it also has similarities with rest gradient 2, with also, again, similar important differences. For example, the default network regions, which is situated between in the midway across this gradient in rest, is now shifted toward the somatomotor end, making it a visual to non-visual, including heteromodal region axis. And gradient 3 was the gradient that has a unique organization during movie watching that was not observed in rest. And it uniquely separates out regions involved in language and auditory regions along one end of that gradient. So if we talk about hierarchical gradients based on the interpretations of these gradients, we know that risk gradient 1 is a hierarchical axis that goes from all the unimodal regions, including somatomotor, auditory, and visual regions on one end, to higher order default network regions on the other end. Whereas if we look at the brain space in movie watching, we see that all of the top 3 movie gradients are hierarchically organized, but they each separate out one modality at the unimodal end. So in gradient 1, we see that it goes from somatomotor to higher order regions. In gradient 2, it's mostly visual regions to higher order regions, and in gradient 3, it's auditory regions to higher order regions. So what we see is that we get 3 modality-specific hierarchical gradients when we characterize these gradients during a naturalistic state, such as movie watching. And hopefully by this point, you can appreciate that using ecologically valid paradigms can tell us something new about the organization of the brain. And to further validate this point or investigate it more, we performed predictive modeling to see if we can predict certain behavioral scores based on gradient features obtained from rest and movie and compare these. So we did a regression model, and we compared movie and rest gradient 1, 2, 3, and all 3 gradients combined to predict the first component of a group of behavioral scores under cognitive, emotion, and motor domains. And we see that we're able to predict cognitive scores, at least with higher accuracy in movie watching gradient-based features compared to rest. We didn't get any significant predictions in emotion or motor scores. So overall, we see that movie watching underscores the hierarchical organization in the brain by describing 3 modality-specific hierarchical gradients obtained during this naturalistic state. And we see that movie gradient features outperform rest gradient feature for predicting, or at least appear to be outperforming rest gradient features for predicting cognitive scores. And this modality-specific hierarchical gradient might provide a unique tool for biomarker research in neuropsychiatry, where we might see unique signature of different disorder on these 3 gradients. So this is still ongoing work, and we want to test the prediction accuracy based on gradient scores versus gradient features versus connectome features, and also look at the reliability of these movie gradients and try to relate it to other structural features across the brain. And hopefully the long-term goal of this line of research is to use movie gradients in neuropsychiatry, to characterize these gradients in neuropsychiatric groups and try to use that in a clinical setting and maybe development of biomarker research. I'd like to thank my supervisor, Tammy Vanderwall, and everyone in the Naturalistic Neuroimaging Lab, including Hallie, who's here, and she has also a poster where she used fMRI from movie watching data. And also, I want to thank Ting Zhu, who is our collaborator on this project, and the University of British Columbia and BC Children's for supporting and funding my research. And if you want to learn more about how we use movie fMRI in our lab, feel free to stop by our posters and have a chat. Thank you for listening.
We get a couple of questions, and only the upvoted question will be speak out here. So if you have more, feel free to chat with Ahmad after the Q&A sessions. Okay, the first question is, what proportion of data did you remove for hand motion, more for rest than movie data?
Okay, that's a good question. So we started with 184 subjects, which is all the subjects we have in the 7 Tesla release. But because of how I talked about head motion, we ended up with 95 subjects for the final stage of the analysis. And that's a small sample size, but we wanted to make sure that at least we're sure that none of the findings we have are, or at least we can minimize the effect of head motion on that.
Okay. The next one is, is before about like why you exclude the force gradients, because it seems like the force gradient is also different between rest and movie, and why only use the first 3? And what about the higher order gradients?
That's an excellent question. Actually, just for the sake of time here, I focused on the hierarchical gradients and just to make sure that this point gets through. But actually, gradient 4 in movie was very similar to gradient 3 in rest. So we see that this gradient 3 in movie is a new gradient that is not described during rest, but then when we get to gradient 4 in movie, it's actually very similar to gradient 3 in rest. And we didn't try to interpret lower order gradients in movie, but that might be something interesting that we can look into. Thank you.
Okay. It's very competitive in the Q&A because like I see like questions keep swatch the ordering because people like upvoted more. Okay, now the top one is why make use of the very fine grained 1,000 parcels? Because it seems like other work may use parcellations with 300 or 400 RIs.
Right. So actually, we did the 400 parcellation and the 1,000 parcellation. It's easier to appreciate the organization, at least in the 3D gradient space, with the 1,000 parcellation. But I think we get similar results when we also use the 400, and that's something that we can provide when we publish the data. Thank you for that.
Okay, last question. Which metric of FC did you choose and why?
Sorry, can you repeat that?
Which metric of functional connectivity did you choose and why?
So we started by the group average functional connectivity matrix in rest and movie separately for the characterization of gradients. And then we computed this new affinity matrix before entering that into the dimensionality reduction analysis. I'm not sure if that answers the question. Or...
Okay. You can always like chat after the session.
Yeah, please feel free to stop by the poster and we can talk about this more. Thank you.
Let's welcome next speaker.
Thank you. So for the next talk, we're going to go clinical with the Merit Award winner, Tobias Bauer, on intrinsic resting state fMRI connectivity on the isolated and connected hemispheres after hemispherectomy. Tobias.
First of all, thanks a lot for the introduction. Thanks you for the organizers for the opportunity to present our data on functional connectivity in patients after hemispherectomy. So first of all, the study group we have at our department consists of 28 patients after hemispherectomy. We just talked about small sample sizes. This seems to be like a small sample size, but given that this condition is very rare, it's basically a large cohort. For those who are not familiar with that, hemispherectomy is a surgical therapy in patients with epilepsy that have an epilepsy which is located to one hemisphere, like in porencephaly or in Rasmussen's encephalitis. And just to make it clear, the difference between functional hemispherectomy and callosotomy, like patients with split brain, is that there is one hemisphere that is completely disconnected from the entire rest of the central nervous system. So that's a very rare and very special condition that we are looking at here. Our patients were recruited between immediately after surgery and 20 years post-surgery, which is a very large time span. And we have two very special cases in that where we had acquired fMRI data before surgery and even have two scans post-hemispherectomy. So to show a little bit or to give you an idea what the brains of those patients look like, I have brought the structural T1 scans of those patients. And if you have a closer look, you can see that in some patients, like in the top row, the hemisphere that is affected by the epilepsy is almost non-existent. So there's not much tissue left on the isolated hemisphere after surgery. The more interesting cases I've depicted in the bottom row are those patients where on the isolated hemisphere there is still a considerable amount of brain tissue left. So we were very interested in including this very special anatomical situation in our analysis. Let's follow up with the image analysis that we have conducted. We had three Tesla T1 weighted structural images where we drew manual lesion masks and applied a volumetric parcellation in the Schäfer atlas with 200 parcels per hemisphere. And we discarded all parcels where 50% all voxels had an overlap with the lesion masks. Next, if we had patients where in all of the 7 networks from the Schäfer atlas, less than half of all parcels were discarded, we analyzed both hemispheres, so the connected and the isolated hemisphere, and that was the case in 9 of our patients. In the remaining 19 patients, we looked at the connected hemisphere only. In addition to that, we have 3 Tesla 12-minute resting state fMRI scans that we pre-processed through fMRI prep and performed connectivity analysis with NILEARN. So next, let me give you an outline of how I will guide you through the results of our study. First of all, we tried to show that this parcellation that we applied on those brains that are very lesioned is actually applicable. As we have shown that, we will look into the connectivity and will quantify it in comparison to healthy controls and also over time. And last, we will have a look at network efficiency before I will come to the conclusions. So then let's jump into parcel homogeneity. This is a little bit a technical part from the study. We have decided to look into 3 different correlation classes on voxel to parcel correlation level. So first of all, within parcel correlation is the correlation between every voxel and the average time series of the parcel that it is assigned to. Next, inside network correlation is the correlation between a voxel and any other parcel that is inside the same network. And lastly, outside network correlation is the correlation between a voxel and the average time series of parcels that are in a different network. So as depicted here, what one would expect is that the correlation is always highest within the parcel that the voxel is assigned to, obviously, followed by inside and outside network correlation. We've calculated this for all hemispheres and all parcels, and as you can see in this graph, this seems to be the case for all our hemispheres, and to make this even clearer, we calculated the average per hemisphere and calculated the difference within minus inside and the difference inside minus outside, which are, as you can see, greater than 0 for all hemispheres, which led us to conclude that this parcellation with the pattern showing within greater than inside, greater than outside network parcellation is applicable in all connected and isolated hemispheres. So then we looked at the connectivity on parcel level. So we computed the pairwise correlations between all parcels. Here I would like to refer us to a study that was published in 2019, where also the authors looked at 6 patients with a quite similar condition, patients with a single cerebral hemisphere after a full hemispherectomy. So the difference to our study is that in those patients, the hemisphere that was affected by epilepsy was completely removed. So they only were able to look at this one remaining hemisphere that corresponds to our connected hemisphere. What they have seen is that there is an increased communication between networks as compared to controls, and this fully aligns with our data as shown in the graph on the left side. In addition to this, we have data to the isolated hemisphere, and there we see that between network connectivity is even further increased on this isolated cortex. We'll look deep into that to see if between network connectivity is specific to any pair of networks, or if this is a very general effect. The comparison between the connected hemisphere and controls shows that this is quite distributed across all pairs of networks. More interesting, the comparison between isolated hemispheres and controls, as seen on the right side. Here we can see that the bluish part are those those pairs of networks where there is a very much stronger correlation on the isolated hemisphere than in controls. And this is mostly limited to this bottom left corner of the connectivity matrix, which corresponds to the connections between unimodal and transmodal networks. We were also interested in looking at those networks, how the connectivity develops longitudinally. Here we see that with a mixed effects model, since we have multiple time points in some patients, here we see that both within and between network connectivity increase over time, both on the isolated and connected hemisphere, but this effect is stronger in the isolated hemisphere than in the connected hemisphere. Here also we were interested in looking at if this is a specific effect to some network or if this is a very general effect. And here I will stick to between network connectivity. And we calculated the same longitudinal model for all networks separately, like shown on the left side. And then we compared the regression slopes across all networks. As we can see, this pattern that there is more of an increase in the isolated than in the connected hemisphere can be seen in the graph, and it is valid for all networks except for the visual networks. And then we compared the slopes, and here we see that on the isolated hemisphere, there is more of a reorganization, so there is more stronger connectivity, especially in the transmodal networks, while there is less increase in unimodal networks. I also show the specific cases where we have two patients that had the functional scans before surgery and at 2 time points immediately after surgery and then again 6 months after surgery. And here I only show the default and the control mode, so the most transmodal networks, which is very interesting to see because we have 2 patients. Patient A is shown with the triangles down, patient B has the triangles up. And if we look at the default mode network, for example, in patient A, we see that on the connected hemisphere, the between network connectivity decreases after surgery, but then increases already in the first 6 months. And even in the isolated hemisphere in patient A, the connectivity increases, and it is after 6 months even higher than it was before surgery, which I think is a very interesting finding. Next, as we have looked at the network connectivity, I will jump into network efficiency. Here we have basically calculated global efficiency and local efficiency as 2 network measures. We can see that global efficiency is decreased in the isolated hemisphere, while in the connected hemisphere it is almost like in healthy controls. And this pattern is also seen across almost all networks with regard to local network efficiency. We have again looked into the longitudinal model. There we see an increase in global efficiency both in the connected and in the isolated hemisphere. But here again, this effect is stronger in the isolated than in the connected hemisphere. And I've shown again those 2 patients with very interesting data. And here again, patient A is the patient where global efficiency decreases immediately after the surgery, but then within the first 6 months, it almost comes to the point where it was before surgery. Again, looking at the slopes for local efficiency in the longitudinal model, here we see that in almost all networks, there is a stronger increase again on the isolated than in the connected hemisphere. With that, I will come to the conclusions that we drew from this study. First of all, we have shown that this very standard parcellation is applicable after this very severe condition in the connected and isolated hemispheres after hemispherotomy. Next, we have shown that there is a functional reorganization in terms of increase in both global and local efficiency over time and increase in global connectivity between networks in both hemispheres after the surgery, but this effect is even stronger in the isolated than in the connected hemisphere. Third, we have seen that there is a stronger remodeling in transmodal than in unimodal networks. And now the fourth conclusion teases something that our collaborators that work into neurophilosophy are very interested about, which is an interpretation that this pattern that we have shown is a necessary, though not a sufficient condition for an island of awareness that is given on this isolated cortical hemisphere. With that, I would like to thank all those who have contributed to the study, the Translational Neuroimaging Group in Bonn, especially my supervisors, Theodor Rüber and Professor Surges. If you're interested in learning more about epilepsy, we have a couple of posters today. If you have any questions, feel free to stop by. I will be around as well.
Thank you very much. So there's 3 themes in your questions. The first one is a debate about your dark and your bright slides. there's two opinions on that. But coming to the scientific questions, there were many different questions about using an atlas on those clinical cases and the severe changes in the anatomy, and how good you would consider the match between the atlas and the clinical brains, and also if the Schaeffer postulation could be used.
I mean, we refer to a study a lot that was that I've shown that used the same atlas as we did, and we decided to try to use this atlas because our idea was first to show that something that is very well established and that has been worked a lot is applicable in this also in these lesion brains. It's true that especially the pre-processing was a little bit difficult due to this very large lesions. And I must admit that we have been in some terms very liberal, but it was still our aim to show that, to at least as much as we could show, that this postulation is applicable. I mean, it's true that some of our next step will be to show, I mean, this is like a top-down approach. We show that something that is established can somehow be applied also on those brains. But we are now aiming at showing, at more elaborating on how from the data, maybe there are a little bit different connectivity patterns that emerge from the data and that are not put like a basket on the data.
Okay, so would that also then include individualized postulations for each patient, or still an atlas kind of approach?
I think sort of both. I think maybe an atlas will be, so yes, we're trying to move away from an atlas, yeah. Okay.
So there was like 5 questions about that, so if you want to have more detail, feel free to find them afterwards. Another common theme in the questions was the absence of behavioral measures. So we know that that is quite an invasive surgery. That's why it's only performed in very specific cases where the patients are resistant to any other treatment. Have you measured any behavioral changes in the long term? Have you seen any particularities after separating the hemispheres?
Yeah, so there are basically, of course, there are lots of behavioral changes. Also because the group is very heterogeneous, so in some patients there are many changes, in others less. But that's basically work in progress. So we have a lot of behavioral measures and also lots of follow-up data. And we're still, I mean, this is just the first steps of our study where we wanted to show that there is some interesting connectivity pattern on the isolated hemisphere. And we're now looking more into how those measures or how other measures that we're working on relate to the behavioral outcome and the behavioral measures, and basically, yeah.
So it's coming, great.
Yeah, we have a lot of data, yeah.
Another question was if any of these effects were related to handedness.
We haven't looked at that yet. That's also coming with the behavioral data, I think. Yeah. Okay.
Then a very prominent question is, were the hemispherectomies that you showed total disconnections? So was it the corpus callosum including also the smaller commissures, or was it partial hemispherectomies?
Those were all total hemispherectomies, so there was a total disconnection, yeah.
Okay. There was another question I wanted to pick up. Did I delete it? Oh, maybe. Here's another one. Are there many ways to quantify functional, there are many ways to quantify functional connectivity. Why do we use Parsons correlation in most of the cases?
I think because it's the most applied measure, so it's the most straightforward measure, so that was the one we started with, I think.
Fair enough. I think I'll let you off the hook now, but if you have more questions, find them afterwards. Thank you.
Now we have the next speaker, Yezhou Wang from the McGill University, Montreal. Let's welcome the speaker.
Thank you for the introduction. So, hello everyone, I'm Yezhou Wang from McGill University. It's my great pleasure to introduce my work here about long-range functional connections, mirror and link microarchitecture, and cognitive hierarchies. So one principle of the human brain is functional integration of large-scale networks that realize through long-range connections. Taking advantage of multimodal neural imaging data, we could construct brain networks. Although most connections in the brain network are short-range, a small number of long-range connections play a crucial role in integrating information. A previous study found that there are rich club hubs connecting to more brain regions, which is important to the brain network. And another study found that rare long-range connectivity is crucial to significantly improving information processing and susceptibility. By the other hand, another principle of the human brain is functional segregation, which likely relates to variations in cortical microarchitecture. Multi-scale methods provide us opportunities to assess brain microarchitecture underlying these principles. For example, taking advantage of neuroimaging data such as myelin-sensitive MRI, we could approximately assess patterns of myelination. And for another example, recent open access rather high resolution three-dimensional mode of human cytoarchitecture provides microstructural properties and considerable neuroanatomic insights into the human brain. And in addition, the gene expression data of the whole brain has been constructed, providing us molecular level insights into the brain organization. So thanks to these multi-scale methods, in this work, we want to address how does long-range connectivity reflect cortical microarchitecture. To figure out this question, we examine the human brain at multiple scales, including microarchitecture and macro-scale organization, especially the long-range connectivity. So in the main analysis, we studied mixed data sets, including 50 healthy and related adults. All participants have quality-controlled T1-weighted MRI, quantitative T1 MRI, and resting state functional MRI. And we also studied big brain. It's a three-dimensional human brain model, as we mentioned before. It's based on a reconstruction of histological sections from a 65-year-old male. And to validate our findings, we studied HCP data sets with 200 healthy adults. So we first estimated connectivity distance coefficient, CDC. We first estimated connectivity distance by taking average of geodesic distance within functionally connected regions. And then CDC is defined as the Pearson's correlation coefficient between seed region's functional connectivity and connectivity distance. So as a result, CDC regions with higher value in CDC means that these regions are more related to long-range connectivity subnetwork. And regions with negative value in CDC indicates that these regions are more related to short-range connectivity subnetwork. When stratifying CDC to an atlas of functional zone, we found highest CDC in heteromodal network and the lowest CDC in idiotypic network. We also estimated the principal functional gradients, and we found a significant correlation between functional gradients and CDC. Next, we assessed the associations between long-range connectivity and the cognitive terms. We performed meta-analysis based on 24 cognitive terms from neuroscience, We found that cognitive terms associated to long-range connections involved in higher-order cognition, such as social cognition and emotion, and while short-range connections are more related to sensory functions, such as multisensory and auditory. And we also assess the associations between long-range connectivity and local cortical microarchitecture. We first studied the quantitative T1 relaxometry. We constructed several equivolumetric surfaces and sampled the intensity profiles, intensity of these surfaces. And then we cross-correlated the nodal intensity profiles to generate the microstructure similarity matrix. And based on this matrix, we estimated the principal gradient and found a significant correlation between the QT1 gradient and CDC. Then for big brain histology data, we also constructed several equivolumetric surfaces and sampled the intensity profiles of these surfaces. But what is different, as we have done for QT1 relaxometry, here we calculated skewness based on the nodal intensity profiles of the big brain histology. Here, higher skewness indicates higher laminar differentiation of the cortex. And as a result, we found a significantly negative correlation between big brain skewness and the CDC. And that means brain regions more related to long-range connectivity subnetwork are with lower laminar differentiation. And to further explore the relationship between microarchitecture and long-range connections, we studied the microstructure similarity matrix of quantitative T1 and big brain histology data. We found a significant quadratic relationship between microarchitectural similarity and CDC, both for QT1 data and histology data. That means long-range functional connectivity may link spatially distant but microarchitecturally similar cortical regions. An overarching mixed effects model aggregating QT1 and big brain histology data confirmed a strong quadratic effect. And finally, all these findings in the main analysis are reproduced and validated on HCP dataset. So, the conclusions. Overall, these findings provide evidence for centrifugal topography of functional connectivity distance and show us a spatially covariant microarchitectural axis. Long-range connections may allow for integration of spatially remote but microarchitecturally similar areas of association cortex, and thus potentially contribute to parallel and higher-order information processing that is important to the human cognition. And finally, I want to thank my lab, my funding resources. I want to thank my supervisor, Boris Berhardt and Alan Evans. I want to thank all of my colleagues and collaborators. Thank you for listening.
Thank you very much. So we have a couple of questions for you, starting with, did you apply any threshold when you computed the CDC metrics? And if yes, will CDC be very sensitive to that threshold?
Oh, yes. Actually, for the estimate of CDC, we set a threshold. We retain the top 10% of functional connectivity, and we use the top 10% of functional connectivity as a mask and apply this mask to the geodesic distance matrix. And we take an average of geodesic distance within this mask. And for the results, if we apply top 10% or top 5% or top 15%, the results didn't change so much. So it's not, the results is similar, yeah. It's not so sensitive to the thresholds of top 10% and top 5, top 15. But when the threshold is top 20, the results may be different. Yeah. So that's why we choose top 10%.
So you showed the Big Brain Atlas, which is obviously based on a single case. How would that impact the generalizability of your results? What's the influence of inter-individual variability given that you compared it to the Big Brain Atlas?
Yes, sorry, could you please?
Sure. So you showed the Big Brain Atlas.
Yes.
That is based on one person.
Yes.
So how would that impact given the amount of inter-individual variability in the brain that we have seen also in the keynote lecture this morning? How would that impact the generalizability of your results?
Oh, yes, the generalizability. Yeah, that's one, that's an issue because there is only 1 mile for Big Brain histology data, but there is a new data named the Big Brain 2, and in the future we may have more data from different donors, so we can further explore whether the results is consistent using the new data. But in this work, yes, it's an issue. It's a limitation in this work. Yeah.
Along the same lines, I guess, the question is, what's the age range in the current sample, and have you applied the same method to developmental data sets?
Development data set?
Yeah.
In this work, no. Yeah.
Not yet.
We don't. Yeah, we don't apply it for the.
And
development
the age range in the current data set?
Sorry.
What's the age range in the current data set?
Age range.
Yeah.
Age range.
Yeah.
Oh.
How old were they?
Is from. I don't remember the exact number, but it's adults. Their sets from about 20 to 45, and the average age is about 32 years old.
Thank you.
Yes.
Moving on to the next speaker. Thank you very much.
Okay, thank you.
So for our next speaker, we have Hoyoung Soon from University of Chicago.
Hello, everyone. This is Hoyoung Song from the University of Chicago, and the work that I would like to share with you today is neural dynamics in a low-dimensional state space reflect cognitive and attentional dynamics. Every day, we experience a variety of cognitive and attentional states that fluctuate on a moment-to-moment basis. Along with our changes in mental states, we know that the brain states also change dynamically over time. In this study, we conceptualize neural dynamics as a trajectory within a state space. A large amount of work showed that neural dynamics operate on a low-dimensional manifold given the covariant nature of the brain. Here, because we are conceptualizing whole brain dynamics, the dimensions here are the activity of the parcellated cortical networks and subcortical regions. Studies have shown that neural dynamics are not just a random continuous flow in the manifold. Rather, there exist several latent states or hidden clusters in the space. Each of the latent states can be characterized by their mean activity patterns and covariance across functional networks. A method that we can apply to infer these latent states is the hidden Markov model, which infers the sequence of the hidden states given the observed brain activity patterns. A fundamental question that we'd like to address is whether the neural state dynamics reflect cognitive and attentional dynamics. There's surprisingly lack of evidence on this because studies have mostly been using resting state fMRI to understand neural dynamics. A more recent work has used cognitive task paradigms like working memory task to characterize neural state dynamics relevant to participants' task performances. another line of research characterized large-scale dynamics in a more naturalistic paradigm like movie watching. However, how do we know that we are not just capturing neural states that are specific to each context? How will we know that they would generalize? Let's think about this further in a state space. I illustrated neural dynamics of cognitive task in green and naturalistic movie watching in purple. Do neural dynamics operate on two separate latent subspaces? Do neural dynamics traverse latent states that are specific to each context? An alternative hypothesis is that there may be this shared latent subspace where generalizable and canonical latent states characterize neural dynamics even when these are in different contexts. If this alternative hypothesis is true, and the brain states indeed generalize, then what functional roles do they play in these different contexts? To answer that question, we collected human fMRI dataset. First was 2 runs of the resting state scans. Second was a controlled sustained attention task called Gradual Onset Continuous Performance Task, or GRAS-CPT. Next, we collected naturalistic task where a participant watched comedy sitcom episodes, which was really fun and engaging. And we also collected educational documentary watching session where a participant watched educational documentary describing history and geographical facts about Korean rivers, so it was relatively boring. We applied the hidden Markov model on the dataset that we collected. We asked what latent states underlie neural dynamics in risk, task, and naturalistic contexts. These are the activity patterns and functional covariance of the 4 latent states that was inferred. The hyperparameter for number of states was decided in a data-driven manner. We labeled the states the DMN, DAN, and SM state based on the functional networks that were most active in these states. Interestingly, the base state exhibited activity levels close to the mean, and their covariance strength was also small, mostly near 0. These 4 states were replicated with another dataset. They were reliable, and they were robust to hyperparameter selection and pre-processing pipeline. Please visit our poster session to know further. With these 4 latent states, we asked if there exists a shared latent states that encompasses all of these states. We were greatly inspired by the seminal work by Margulies and colleagues, which characterized main gradients of variance in the resting state functional connectome of 820 individuals. This study showed that low-dimensional gradients of functional brain connectome captures the unimodal to transmodal axis, as well as sensory to motor axis of the neural systems. We situated our 4 latent states onto the spatial gradient axis based on their activity patterns. First, this is the position of the DMN state on the gradient space, the DAN state, the SM state, and finally the base state. You can see that the 4 brain states tile the extremes and the center of the gradient space. A large amount of variance was explained by the predefined gradients, and we observed that this base state, which located at the center of the gradient space, acted as a hub in neural state transitions, such that neural dynamics were more likely to transition to the base state when making transition to another state. These results propose that the neural dynamics can be understood as a traversal along the canonical gradients of functional brain organization, which operates as a common latent manifold across diverse contexts. So far, we observed 4 latent states that were shared across multiple contexts. Now, we ask whether the latent state dynamics or the trajectory along the state space differ across contexts. Here, I listed 7 runs of the dataset, and this is the latent state sequence of the 27 participants that were collected, with the color indicating one of the 4 states that occurred at each moment of time. The distributions on the right indicate the probability of latent state occurrence. This is the latent state sequence of the GRAS-CPT runs, the sitcom episodes, And finally, the documentary. You can see that the state occurrence patterns differed across conditions. However, they were very similar among runs of similar contexts. This means that although the 4 brain states commonly occurred throughout contexts, the probability to which they occurred differed across contexts. Furthermore, we observed that the latent state dynamics were highly synchronized across participants during sitcom episodes. This means that neural state dynamics reflect participants' cognitive state dynamics. But then exactly how are the neural state dynamics being modulated by stimulus-driven cognition? To understand this further, we made use of the fact that our sitcom stimulus had clear event boundaries such that 2 separate events happened in an interleaved AB-AB sequence. We asked if any of the latent state was more likely to occur after event boundaries. The figure here shows the occurrence proportion of the DMN state and the base state time aligned to narrative event boundaries. The gray areas here indicate the null distribution, and this is the actual proportion of the DMN state after event boundaries. You can see that the DMN state increased after event boundaries, whereas the base state decreased this occurrence, compensating for the increase in the DMN state. This result was replicated when we applied the model to the external dataset. We statistically observed that the neural state transitions directly to the DMN state at event boundaries, breaking this normal tendency of having to pass through the base state in order to reach the DMN state. This means that neural state dynamics are adaptively modulated in response to the external cognitive demands. Finally, we asked whether neural state dynamics reflect participants' ongoing changes in attention. During GRASS-CPT, the stability of participants' response times indicated whether they were attentive or inattentive at every moment of time. For sitcom episodes, we had participants come out of the scanner, we replayed the video, and asked them to continuously rate how engaging the stimulus was at every moment of time. We related participants' behavioral measures with neural state dynamics by averaging the attention levels at moments of latent state occurrence. This is the result of the GRASS-CPT runs. You can see that participants were highly attentive during the DMN state, whereas they were inattentive during the DAN state. This means that DMN state reflect high attentional engagement during controlled task performances, which conceptually replicate past studies. Interestingly, we observed a different relationship between attention and neural dynamics during sitcom episodes, such that base state indicated high attentional engagement to narratives. These results were again replicated when we applied the model to the external datasets. Furthermore, an interesting observation that we had was that this SM state indicated inattentiveness in all of this context except for a single GRASS-CPT run. Together, though different neural states indicate high attention in different contexts, it seems like the same neural state indicates low attention in both task and naturalistic conditions. To conclude this talk, we used the hidden Markov model to find the common neural states that underlie diverse cognitive contexts. The latent state dynamics span canonical gradients of functional brain organization. Lastly, using both controlled and naturalistic tasks, we showed that the neural state dynamics reflect changes in cognition and attention. I would like to acknowledge my co-authors/mentors, Dr. Monica Rosenberg and Dr. Won-Mok Shim, and we also thank many researchers who open sourced their data and code, which greatly helped our project. Thank you.
So in the interest of time, we're going to go with the top questions, but feel free to have a chat after this session. Can you explain how you mapped states to the gradient spaces?
Yes. So how we did it was Dr. Margulies and colleagues open sourced the gradient values of all of the voxels that tile the entire cortex So we first averaged all the gradient values per parcels, and then with the activity patterns that were inferred per state in our HMM analysis, we did element-wise dot product between the activity patterns and the mean gradient values of the parcels. And then from that, we mapped the mean element-wise product to the gradient space.
Lovely, thank you. That's all the time we have for the session, I'm afraid, but feel free to find the speakers in the lunch break or over coffee for the rest of the day. And thank you all for being here. Thanks to our speakers.
Thank you.